So that's the viral structure. And then when we're looking at the viral structure, if you just look at that on the top left, this is a structure. On the right bottom right here, this is a visualization from the electron microscopy. And these are all based on structure. So if we do not understand the structure, it is almost impossible for us to understand any of the physiological processes overall, and the mechanism of the virus, that how these molecular nanomachines actually do work. So it is an essential and integral part of it. I do not have to elaborate that. I have just written down why it is. And even for the therapeutics development, that is also essential. Even for vaccine development, it is also essential, because now people actually know what it is. And if the virus is mutating or something like that, we can map those things. Particularly, that has been done extensively for the influenza virus and so on. So without further discussing that, but you can just read the narrative of the opening slide. Let's go into the material. So the first thing is that what is the role of these structural proteins which actually make up these viruses? So the first very important thing is that protection of the genome. So the genome is inside right here, which is cut open in this influenza virus cartoon here. These are now enclosed, but you know the bacteriophage, the DNA is packed inside the head. This is a small, like a parvovirus, which is a small molecule of DNA, which is packed inside. So essentially, that assembly of a stable protective protein shell is important, in which the virus protects the genome. Why it needs to protect the genome? Because remember that in the environment, they can be encountering lots of harsh chemicals. Not only the harsh chemicals, many enzymes, these proteolytic enzymes, are everywhere. Nucleases, which can chew up DNAs or RNAs, are everywhere. Even our hand, like a palm of our hand, contain a huge amount of these nucleases as well. So those are some of the protective features. So these structures need to be protected. And that's the reason of the structural proteins, protection of the genome. And also along with that, there are some specific recognition and packaging of the nucleic acid genome is also related. So what we mean by that when these particles are being synthesized, there are structural protein motifs or topology on these that will actually allow the genome to be packaged or put into the box, that we say that word box all the time. So think about that in a package assembly line in a company or something like that, each box is marked that what will go into that box. And this is how the viruses does as well. So they are marked for the specific recognition on packaging, and we'll talk about that in a separate cases. And also it will interact with the host cell membrane to form the envelope. You can see that right here, this structure is a naked structure, completely protein. Although the bacteriophage may have an envelope underneath this structure that you do not need right now. But on the other hand, right here, you can see that there is a viral envelope outside. So this is an enveloped virus. So the second function of the structural protein is the delivery of the genome. And here you are looking at a bullet-shaped rhabdovirus, which is a reovirus. Here you're looking at a pox virus. And this is also another single-stranded RNA virus. Here you are looking at enveloped all of them. And the important is the delivery of the genome. To do this, it needs to bind to the host cell receptors, or it may fuse with the plasma membrane as well. So this interaction has to be done. And then it also allows to uncoat of the genome, fusion with the cell membrane, as I said, and transport of the genome to the appropriate site, either the cytoplasm or to the nucleus. And remember that just entering the cell is not good enough. Once the viral particle enters the cell, usually it will use some kind of a vesicular transport to transport that genome to a target site. And this could be microtubule mediated. If you have taken cell biology, you already know that big vesicles can be transported. This could be inside a vesicle, and that whole vesicle is transported by motor proteins on the microtubule highways inside the cell to deliver the genome. But if you just remember, transport the genome to the appropriate site, that is good enough. So these are the two main classes or function of the structural proteins. So the important thing is that we already told that the virus particle must protect the genome. And this tells you that it is a stable function. This is the first slide. The second slide tells you that delivery of the genome, when it delivery of the genome, it also need to uncode the genome. So the genome from the inside need to come out. So it must come out in the infection. And this is a unstable structure. So how does a stable structure become unstable? And this is achieved by what we called in virology as the metastability of a virion. So metastability means under certain conditions, the virion is extremely stable, but it can come apart if there is a proper signal given. So here you can see that a particular virus is attaching to a receptor on the cell surface. Right here is a membrane, and this is the cell. These are the cytoskeletal elements. Once it is inside this endosomal vesicle right here, there is a signal provided into this, and we'll talk about those in detail in attachment and entry. Once this signal is received, the particle becomes unstable, and it will uncode to deliver the genome. So this is what we mean by the metastability. So how this is actually achieved? So the viruses, for getting apart or taking apart, they have not retained or attained the minimum free energy conformation. So what happens that a virus which is stable in this state, which is number 1 state. So the number 3 state, which is a minimum free energy, see that right here, this is a lower energy. This has not retained this because it did not receive that signal. So what happens to go into this form, which is unstable, the minimum free energy conformation, it needs to go over a hump before it can do this. So how it is obtained? Before coming to that, what happens that once the viruses are being made, energy is actually put into the virus particle during the assembly, which is called the spring-loaded. And you all know that this is the bonding energy. The bonding energy is put into the virus, which you can think about as being spring-loaded. Now, in all conditions, it is very stable. In certain condition, it can go over this hump, and this hump going over this is achieved by the interaction of the receptor and the viral particle. Once it goes over that, it can attain the free energy conformation, and spontaneously, this structure is disassembled. And this potential energy, which you are calling about the spring-loaded, the bonding energy, are used for the disassembly if the cell actually provides the proper signal. Just for reality, that what happens, suppose a receptor binding is taking place, that will change a conformation of a protein in a local area, and that will spread like a fire across that. It will change a global conformation of all the protein molecules across the viral particle, and the virus will come apart. It just need to start at one area, and that one area will be just going over that hump. So it is spontaneous. So it is an amazing nanomachine. It is just not an inert structure in that sense, that it is just a molecular nanomachine that works under some chemical signals. So how it is achieved? That it is created by the symmetrical arrangement of many identical proteins to provide maximal contact. And these maximal contact is achieved by an unstable structure, in the sense that these bondings are not covalent. They are not permanently bonded together. But the arrangement, or the symmetrical arrangement, or the interaction of these proteins are usually hydrogen bonding, electrostatic bonding, hydrophobic interaction, van der Waals force, and they are much more easy to break if they are not a covalent or a permanent bonding, and can be taken apart, loosened on infection to release the genome. So this just a cartoon right here actually just shows that how easily you can achieve this, that you are looking at this rhinovirus model, which actually shows you what is being shown here. This is a rhinovirus, and again, the detailed names are not important, but you can see the different proteins are colored in the blue, red, and the green, and just this is a three-dimensional topology, and you can see that there is a close interaction. There is no empty space in between them. They are bonded just like head to head, tail to tail. And these interactions are what I mean to say is not covalent, but these are usually hydrogen bonding, electrostatic interactions, hydrophobic interactions, and all these different types of interactions, which can be easily taken apart in that process as well, which actually gives you the metastability. All right. Being said that, let's go to some of the definitions, a very important slide. So the first definition is what is a subunit in a virus structure. The subunit is a single folded polypeptide chain. And remember that a single folded polypeptide chain could be just a protein. And this single folded polypeptide chain is the tertiary structure of the protein. And just to recap your initial biochemistry or biology material, that you have the amino acid, just the sequence of the amino acid is your primary structure. And then if you just fold them into alpha helices and beta sheets, they are the secondary structure, and the collection of the alpha helices, beta sheets, and the folds or the loops actually make them the three-dimensional structure, which is the tertiary structure. If many protein subunits actually join together to form a larger protein, like say hemoglobin or something, that's called a quaternary structure. But here we are looking at that they are the folded polypeptide chain, individual protein molecules that is being built. So here is an example of a poliovirus. And here the capsid is made up of 3 proteins, so VP1, 2, and 3, and each one of them is a polypeptide chain. So you have 3 subunits in here. The structural unit, which is also known as protomer or asymmetric unit, is the unit from which the capsids or nucleocapsids are built, and this could be 1 or more subunits. So you can see that if you just look at this triangle, which is in blue, this triangle is made up of 1 blue, 1 red, 1 yellow, and all these 3 together is the structural unit or protomer or asymmetric unit. Now, remember that instead of these 3 different proteins, that is a very, very important idea. Instead of three different proteins, a capsid can be made up of 1 protein, just 1 protein, but it has to be in this conformation. It has to be a trimer. You have a monomer here, you have a dimer here, and you have the trimer. So 3 monomers can build the same structure out of a same single protein. But it can get more complicated if you use more and more, and we'll come back to that. The next is the capsid that we constantly call as a box. This is the protein shell surrounding the genome. You can see that this is a naked TMV, or the tobacco mosaic virus. It contains just only 1 protein subunit, which encloses the RNA, and this is a naked capsid that is being shown. The nucleocapsid, on the other hand, is the genome and the capsid assembly within the particle. And sometimes people get confused with this. There's no confusion. It's very simple forward. If this RNA and the capsid is together present, we can call this as a nucleocapsid. If you take the RNA out and there's an empty capsid, then it's just a capsid, not a nucleocapsid. Now, so why we use the capsid and the nucleocapsid? Just to use this term specifically, say for an example, in the assembly of the virus. So we can state a term like this, that when the virus is actually assembled, the nucleic acid is put into the capsids, which means empty capsids, and forms nucleocapsid. That means after the nucleic acid is put into the capsid, it becomes the nucleocapsid, which could be either enveloped or naked. This one is naked, whereas right here, this is a nucleocapsid in the middle, right here, nucleocapsid, which is surrounded by a membrane. And this is a discrete substructure in the enveloped viruses. An envelope is the viral membrane, and this is a host cell-derived phospholipid bilayer, and does not necessarily mean that this is a part of the plasma membrane. Remember that this envelope can be derived from the endoplasmic reticulum, from the nuclear membrane, but any kind of membrane that is present in a cell. And the virion, we already know that infectious virus particle is the definition. So how we study the viral structures? So electron microscopy actually paved the way of that, and then X-ray crystallography delivered the ultrastructure of these different viruses. And then lately, the cryo-electron microscopy or the cryo-electron tomography actually gave the the very, very high resolution structure, almost similar to the resolution or even sometimes better than the X-ray crystallography. Now, we are just going to talk about just a little bit because this is very close to my areas, but we are not going to discuss this. Is not a structural biology course. That's why this part will not be required for just for reference or fun purpose. On the other hand, nuclear magnetic resonance or NMR can also solve structure, but only they're restricted to individual viral protein molecules, which are smaller proteins actually. Usually it is difficult to solve a structure by NMR if the protein is more than 20-25 kilodalton. So not a viral whole structure. The whole viral structure can be visualized by the three techniques. So here we are looking at some of the definitions. What is the angstrom is 10 to the power minus 10 meter, and nanometer is 10 to the power 9 meters. So 1 nanometer is equal to 10 angstrom. So approximately the atomic dimension is 1 angstrom. So why X-ray crystallography is used to look at or solve the atomic structure? The idea is the wavelength of X-ray is exactly 1 angstrom. So that can, that's the reason that electron microscope cannot do that, which is restricted to about 50 to 75 angstrom at the most. And that's the reason that using the 1 angstrom wavelength of the X-ray, we can solve this structure. But we cannot build an X-ray microscope. We cannot visualize that. And we can only get an electron density, which is by a process, gives the idea of the location of the individual atoms in which we can build a realistic model. And they are very accurate. What you are seeing right here at the top, these are a couple of amino acids, and each amino acids where you can see that the nitrogens are in the blues and the oxygens are in the reds. And this mesh-like structure, what you see around them, is called the electron density. So this is basically based on the experimental data, which gives you the idea of where the electrons are or where the electron shells or the clouds are. And you build your model inside it because you know the structure of these individual amino acids. So you can trace them and put them in there using some of these programs. And these are some of the overview of those, and not going into the detail of that. So you can see that amino acids are in between 0.4 to 1 nanometer, whereas the alpha helices of a protein right here is about 1 nanometer in the diameter. And this gives you a perspective of that. Say small viruses like poliovirus are about 20, 30 nanometer in size, whereas the giant viruses could be 1,000 nanometer, even 1,500 nanometers would be the size. So let's take a look at the idea that first 1940s, the Helmut Ruska actually invented the electron microscope, and that completely changed that how we study the viruses or the structural biology of the viruses. So you can easily see that this was the first picture that came out, that a bacterial cell and the bacteriophages on top of that. This is the original publication from the 1940s. And this amazing detail was completely elusive before the advent of the electron microscope. And after that, you can see that all these different structures, and most of them are what you are seeing. That structure is a contrasting black and white. You cannot get a colored picture of an electron microscope. If you see an electron micrograph which is colored, it is photoshopped. It is a pseudo-colored, which is on the very first slide or the opening slide that you see on the top right side. That is a pseudo-colored slide. You just only get black and white pictures. So these are all usually negatively stained to contrast, and these are all from your previous other courses, not going into the detail, but this is the overview of the first structural observation of the viruses. But the problem is the resolution actually cannot be pushed at more than 50 to 75 angstrom. That is not enough to look at the molecular level. So the next thing what happened is that how we actually determine the structures at the atomic resolution, the X-ray crystallography. Busy slide, and this is for reference only, just for fun. So what we do that to solve this, first we need to get a crystal. So once we get a crystal, we actually take them to an X-ray source, and many a times your in-house X-ray source are not enough, you go to a synchrotron, which is like this. These are in national laboratories. So this picture is the Argonne National Laboratory, which is 55-mile west of Chicago. It's amazing facilities. And this is the storage ring with a linear accelerator. So what happens, the linear accelerator, which is in the middle, actually inject this high energy electrons into this storage ring. And at certain intervals, like this is the storage ring, and in certain intervals, these electron beams come out, which we call as a beam lines. And when these high energy electrons comes out, it produces the X-ray, which is used for the several purposes. Remember that these high energy electrons can produce different intensities of different things, different radiations, and is used by scientists from material science to chemists, physics, biological sciences, everywhere. And this runs 24/7, 360 days a week. There is literally just a couple of days or hours of downtime, and something that of this magnitude that you can just compare with some space installation of NASA or something like that. It's an amazing facility. So this is just a picture. We used to work in here. When I was at Case Western, we used to go there every month. And this is just how the hallway looks like just inside here. And this was our structural biology beamline 19ID. And this is a picture from 15, 17 years ago that one of the beamline where we used to collect data, X-ray diffraction data. It's an amazing facility. Anyway, so once you get this diffraction data, you get an electron density map, but the problem is that what happens that there is one piece of data is missing in the X-ray crystallography, which is the phases. Like in the just to have an overview of an idea, do not have to go into detail. We really do not know that if the reflection spot or the diffraction is coming from this atom or this atom, because there is a time difference, and that has to be solved by something which we call as the anomalous scatterer. So here you can see the phase problem in this molecule. See that when the incidence of the X-ray is coming right here and it is diffracted in here, this wave front is more or coming faster on the screen, which is recording that, and this one is coming slower. But you are taking an image or shot at every certain seconds. So when you take a shot, you just get a spot here. You also get a spot here. Whether this spot arrived earlier or later, you do not know. It could be just the intensity is more here, intensity is less here, but that does not give you the spatial difference with where they are in the three-dimensional space. We solve this with different types of techniques called MIR, or multiple isomorphous replacement, where we soak these proteins with a heavy metal, and that heavy metal, we know that in the protein where it binds, and compared to a normal crystal which doesn't have the heavy metal, we compared the data with that heavy metal, and that gives you a difference map, and from that we can calculate that. Don't have to go into the detail, but this is just an overview of that, how it is done. And once you get that electron density, you actually build the atomic model inside that, because you can fit in the density each amino acid, basically. So you know the structure of each of these amino acids that we saw right here, and you just have to fit those to build your model. It takes a lot of time, but amazing programs and algorithm works this way. So the next event is the cryo-electron microscopy. The reason the X-ray crystallography is a kind of like a black magic kind of thing. Why it was kind of a black magic? Because the problem with the structural biology was that it may not crystallize, it may not diffract. A lot of these problems used to happen with crystallography, but that is solved by the cryo-electron microscopy, where you take the image and you take a two-dimensional picture of those and you Fourier transform. So these are the two-dimensional projections of these three-dimensional structure, and then you can rebuild or merge them to get an average. Now, in the tomography or the cryo-electron tomography, what you do, that you tilt the image, like the platform of the image, you tilt the image at certain degrees, like 1 degree at a time to plus minus 70 degree. And each picture that you get is a little bit tilted in 1 degree, 2 degree, 3 degree, like those. And you can just make those Fourier transform from those tilted images, and you can get three-dimensional image reconstructed from that as well. So this is a 3D reconstruction and an installation. And you can see that at 20 angstrom, what you can see, and at 2.9 angstrom, what you can see. And in the background, you actually see an electron microscope. So you can see easily that what you can see on an electron microscope versus what you can look at the 2.9 angstrom. You can visit that link to look at some of the viral structures. That is a lot of fun if you wish to. All right, so now we are going into the main thing. So this was just the overview of that. The viral structures can be classified into helical, icosahedral, or complex structures. And that's about it. So first, we are looking at the virus particle. The symmetry is the key, and Watson Crick actually did more than discovering the structure. They actually has an enormous contribution into the virology, and that is actually called as a Crick and Watson, because that was the Crick was the first author in that paper, and they actually talked about an idea of genetic economy. As the virus genomes are small, the particles will be built with many copies of the few proteins because it cannot code for hundreds of proteins. And that is one of the very important factor here. And identical protein subunits are distributed with the helical symmetry for rod-shaped viruses and icosahedral symmetry for the round viruses. So why these symmetries are needed? This is due to this genetic economy. So this is the main concept. So for this is a symmetry rules. These are elegant for their simplicity, and they provide the rules of this viral self-assembly. The rule 1 is that each subunit has identical bonding contact with its neighbor. Repeated interactions of chemical on the complementary surfaces at the subunit interfaces naturally leads to a symmetric arrangement. And these bonding contacts are usually non-covalent. We was constantly saying that this is the most important thing, and these are reversible. That's why when the viruses are being assembled, it actually can do an error-free assembly. If there is an error, it can disassemble and reassemble once again. But we will see that we will see so many anomalies in there, and we will going to talk about that. Although we actually saw that each subunit has an identical bonding contact with the neighbors, and we will see that this, although elucidated a lot of viral structure, that is not true for many viruses. They may not be identical bondings. So first, let's take a look at the helical symmetry in the tobacco mosaic virus. And here you have the coat protein molecules engage in identical equivalent interaction with one another with the viral genome to allow construction of a large stable structure from a single protein subunit. So here we are looking at a schematic. Remember that this is not a TMV example, this is TMV example. Here we are looking at one protein subunit. And here we are looking at 1, 2, 3 subunits. So per turn, you have right here 3 subunits. And this is what we call as the mu, which is the subunits per turn. Then the next definition, what you in the helical symmetry, is called the rho, and this is called the axial rise. So think about that. It started right here, and when it just went into the turn, the top surface of this from this surface raised, suppose this is 1 nanometer up, and it is then the rho or the axial rise is 1 nanometer. So from that, you can actually find the pitch of the pitch of the screw, which is the whole structure. Remember that I actually said a little bit wrong here. So what you will see that the rise from one subunit, one end, from here to here, this is 1 nanometer. Here to here, it is 1 nanometer. Here to here, it is a nanometer. So when it is turning one turn, it becomes from here to here 3 nanometer. That is the pitch of the screw axis that you are seeing right here. So you can easily find out with the formula of the P is rho into the mu, which is 3 nanometer. You may have some simple math problems in your exams or the quizzes. So particularly for the tobacco mosaic virus, you can see that the mu is 16.3 subunits of protein per turn. Axial rise is about 1.4 angstrom, and the pitch is 22.8, or for all other purposes we take it as a 2.3 like nanometer or 2.30 angstrom. Now again, remember that 22.8 or 22.3 angstrom becomes 2.3 nanometer. So just familiar with the thing. So this is your helical symmetry. Now, most of the DNA and RNA viruses with the helical symmetry, they are naked, but there are a good number of viruses which could be covered by an envelope as well. Now, the interesting part of that, these viruses, which are the naked viruses, are only found mostly in the plants or in the bacteria or in the archaea. Some of them are naked, some of them could be enveloped. But there is no example for those non-enveloped helical symmetry viruses which in the animals which are not enveloped. So these are just a couple of examples of that, very self-explanatory. So here we can see that these are the few representative viruses with the helical capsid. And again, as I said, that for reference only, but gradually get familiar with the representative cartoons. And if you look at that, none of these are naked viruses. So again, remember that all helical symmetrical viruses, animal viruses, all animal viruses with the helical symmetry are covered with a membrane, and none of them are naked. That means inside that they have the nucleocapsid. Inside that they have a distinct nucleocapsid. So these are DNA, RNA viruses, plus, minus, ambisense, all different types, including retroviridae as well. So here you are looking at a Sendai virus, and this Sendai virus, this is the core or the nucleocapsid. But in reality, this is actually covered by a membrane, large virus. So all the animal viruses of the helical symmetry are always enveloped. The next, what happens and what is the difference between that? So far, what we were seeing that you have this nucleic acid, RNA or DNA molecule covered by a protein, but sometimes this nucleocapsid protein, you are looking at just one protein, but there could be more protein involved in forming the nucleocapsid. And why it is necessary, that we are going to explain in the next slide, which is an example of a vesicular stomatitis virus, VSV. This is a prototype virus of the Rhabdoviridae family, which is the rabies virus. This is the virus I used to work with, transcription, mainly the transcription replication of this. But this rhabdovirus, or the vesicular stomatitis virus, which is a prototype of it, you are looking at, this is a bullet-shaped virus. And right here is the original raw image from the cryo-electron microscopy, and this is the structural build. What you are seeing that right here, this reddish part is the RNA, which is encapsulated into this green protein, which is known as the N protein. Sometimes these are known as NP, or nucleocapsid protein as well. So this is actually the real nucleocapsid. But this nucleocapsid is put in place by another top coat of protein, which is known as an M, or the matrix protein. And then you can see that this is coating, and this is the envelope, which is not needed, outer inner leaflet of the membranes. But this nucleocapsid is covered by these additional proteins. And here you are seeing the three-dimensional structure of the N protein wrapping up this green. The green is the RNA, and this is just a single N protein. And each of these monomers actually cover 9 nucleotides of the RNA. But what experiment can be done here to understand the role of these M proteins? And this is also can give you an critical thinking questions. I have elaborated that in your notes. So if you take the VSV and you just treat it with a detergent, these membranes will come off because they are phospholipid. And then you can treat them with a high salt, which will remove this M protein. If you remove this M protein, this inner structure, which is called the nRNA, actually become very extended and filamentous. And this nRNA in vitro in a test tube is completely replication competent. You add the nucleotides and the condition, and it will replicate the RNA in vitro. So it retained its functionality in that condition, but just for the packaging into this compact structure, this matrix protein is needed. That is what I wanted to say, that additional proteins are needed for the compaction and the packaging. All right, so now the next idea is that how can you make a round capsid from proteins with irregular shapes? And this is where we are going from the helical symmetry to the one which we know call as the icosahedral symmetries. So here what we are looking at, that all round capsid have a precise number of protein in the multiples of 60s, like 60, 180, 249, 60, like this. The second clue is the spherical viruses come in many sizes, but the capsid proteins are about 20 to 60 kilodalton range. And Watson and Crick concluded that these are built in with the icosahedral symmetry. So what do we mean by this? So if the proteins are in between 20 to 60 kilodalton, as the viral structures are getting bigger and bigger, you have very small like 20 nanometer, but you have very large like a 150 nanometer for say adenoviruses. So how does, okay, all right, we understand the smaller ones, how it is built. You have just one protein and that can build those. But if it is a larger and larger viruses, the only way you can build this structure is by this icosahedral symmetry. And these are some of the ideas that was put forward initially. So we'll just elaborate this for the rest of the good amount of time. So what is an icosahedral symmetry? So icosahedron is a classic in geometry. It's called a Goldberg polyhedra. It's a solid with 20 faces and each with an equilateral triangle. And this allows the formation of a closed shell with the smallest number of 60 identical subunits. So on top of that, what you have, that each one of them right here, what you are looking at is a icosahedral triangle. And this, when you rotate this in different areas, you can get different kinds of symmetry. So right here, if you look at that, if you just rotate, you have a threefold axis, you have a threefold symmetry right here. On the other hand, if you rotate around this axis, you will have 1, 2, 3, and on the back side, you have a fivefold. So you could just look at the fivefold from the top right here. You're looking from the top, you have 1, 2, 3, 4, 5. On the other hand, if you just right here, twofold, you can see that you have a twofold symmetry if you rotate the structure in this orientation. So again, remember that all these icosahedrals do have a 3, 5, and a twofold symmetry. And sometimes it could be a little bit difficult to understand in this two-dimensional structure. If I have time, I will show you some three-dimensional pictures, and that may help you a little bit. Or in the discussion, if we just do it the day before the exam, we can show something. So this is the basic idea that how the virus structures are being built. So a simple icosahedral capsid, where we call that the triangulation number is 1. And we will define this a little bit later. But the simplest one is made up of 60 identical protein subunits. This is absolutely the minimum number that is needed. The protein subunit is the structural unit, and it interacts with all molecules with their neighbors head to head, tail to tail. And the particles are spherical, but not icosahedral, because think about that. Although you have the symmetry, it looks like three-dimensional, but the proteins actually do have a curvature on their top. It is not just a flat, rigid structure that we are seeing. So what you are seeing by these comma-shaped structures, this oranges and the grays, these are the individual protein molecules. So around the threefold symmetry, you have 1, 2, 3 proteins. And on the fivefold symmetry, you have 1, 2, 3, 4, 5 proteins. At the twofold symmetry, you have 1 and 2 proteins. So this gives you the symmetry of the simplest type of icosahedral capsid. But things get complicated quickly. Before that, we are just looking at one example. This is an adeno-associated virus 2, which is a parvovirus, simplest one, just like the triangulation number with T1. And again, we will explain. This is made up of 60 copies of a single protein that we said. And you can easily see that it is very difficult to understand from here. But in this different areas, as you can, if you see from the top, this is the 5-fold axis, these are the 2-fold axis, and this is the 3-fold axis. And these bonding patterns actually do have a particular topology, which is known as a beta barrel jelly roll topology. And you have these beta sheets which are connected with another angled beta sheet structure by some loops. And we call this beta barrel jelly roll, and we will elaborate that. And this actually is a very typical topology or motif which is present in the proteins, or many of these proteins which actually make up these icosahedral particles. So this is an overview of the whole structure that you can see from the cryo-electron microscopy. And right here is just one icosahedral triangular face of one symmetry. It does not look like the exact geometric shape, but you have the symmetry right here. You have a 3-fold symmetry right here. You have a 5-fold symmetry right here, and you have a 2-fold symmetry right here. And again, you do not have to go into the structure, the physics of this or the mathematics of that, because that is beyond this course. If you just have an overview of the understanding, that is good enough. So how larger particles are now made is the problem. You can add more subunits. And these subunits are the pentamers known as pentons, or the hexamer, or the hexons. And there are 3 modes of subunit packing, and you can see that they are in orange. Then you have in yellow right here, and the purple. So here what you happen for the other ones, which you have seen before, all of these units were made up of the same protein. These are all the same protein, all these 3 proteins. Here we are making them with 3 different proteins, different subunits, 3 modes of subunit packing. And what happens, these bonding interactions, we are introducing a term, is quasi-equivalent and all engage tail to tail and head to head. And this quasi-equivalence is observed when each triangular face is made up of similar, smaller identical facets. So what we mean by that, in this structure, in the previous structure, all of these are identical. So the interactions in between this protein, and this protein, they were exactly the same, right? All of them. So interaction in between these protein, these 2, and these, they were identical. Now, if these 3 proteins are different, the interaction of these proteins, these 2 in between AB and AC and BC, are supposed to be identical. And this is known as the quasi-equivalence. And this helps to build a bigger structure. But in reality, these quasi-equivalent, there are lots of exceptions where this quasi-equivalence does not exist. And we will talk about that. But this gives you a very interesting structure. You now have, if you have a structure where the triangulation number is more than 1, not simple, you have a 6-fold symmetry. So if you just look at right here, you have 1, 2, 3, 4, 5-fold symmetry right here. Now you have a 6-fold symmetry, 1, 2, 3, 4, 5, 6. So that is the reason that we told that to build this structure, you have to have the pentamers right here, as well as hexamers, or the hexons of the same proteins. And this is how these structures are being built. So when a capsid contains more than 60 subunits, each occupies a quasi-equivalent position. The non-covalent binding properties of the subunit in different structural environment are similar, but not identical. And I have elaborated this in the narrative, in the writing, so you can easily read that carefully. So now we are looking at that how we find the triangulation number. So to find the triangulation number, we are finding, suppose this is a typical icosahedron, and each one of them is right here. We are looking at the 5-fold, 5-fold symmetry. So we are originating from a center of a 5-fold symmetry. And if we just take this surface vector from one direction, suppose this is the H vector. And if we just go in another direction, this is the K vector. They are just in different directions. Whatever is K and whatever is H is the same. So the main thing is that the H and the K vectors originate at the center of a pentagon. So how your T number is actually calculated after that, what will be the smallest number of steps that you have to cover to go from this pentagon to the next one, the smallest distance? So to calculate the vector required to reach the center of the closest fivefold axis from one another. And the T will give you the formula as an h square plus hk plus k square. And if you just look at this number, the number of subunits will become 60 multiplied by that T number. So if your T comes out as 2, it will be 120. If it is 3, it's 180, and so on. So let's take a look at the T numbers, and you will understand that how it is calculated. So suppose this is a pentamer and this is a pentamer. So you can see that right here, you have the very simple way that how it is. You just go in one direction and then you go into the other direction. So let's take a look at an example of that, which is a T number is actually 4. So how you calculate? So from the center, you are traveling. How you are traveling? You are traveling 1, 2, 3, 4. So you are traveling 4 in one direction, and then in the K direction, you are traveling 1. So that's the most important part. If you have that, what happens in this case, your H equal to 4. 1, 2, 3. So from here, 1, 2, 3, and 4. And K equal to 1 from here to here to reach the closest pentagon. So if the T is h square plus hk k square, so you get 21 is the T number for this. So if you get 21, then what happens? Then your number of subunit is very easy to calculate. You just multiply this with a 60T, it becomes 1260 subunits. And this is how you can calculate that. So this is your triangulation number, and how you actually get that triangulation number to go from one fivefold axis to the fivefold axis through the centers of the pentamer, of the fivefold axis, or the pentagons. All right, so here is another example of different triangulation number and the quasi-equivalence. And this is the structural unit. Here you have the organization at the fivefold axis, and this is T equal to 1. And as you are seeing that it is becoming complicated and complicated, T equal to 3, 4, and 13, how the structure is being built, and they are not icosahedral, but they become spherical. And again, do not have to go into the math too much as such. You can always calculate that from here to here, how you have to travel through the pentamers, but it could get really complicated. So if you want to have fun, you can have, but it is not needed for your exam or the quiz as such. So here we are looking at one of the example, which we call as a T equal to 3 structure. We look at the T equal to 1 structure. So now you understand that what is the T equal to 1 structure right here in the parvovirus. Here we are looking at the T equal to 3 structure, which is a poliovirus. Why it is so? You can easily look at that it is made up of 3 proteins, BP1, 2, and 3. And this is right here is one structural unit, and you have these 3 proteins right here. And this actually shows you the beta-ballotol Jerry roll topology, where you can see that you have these anti-parallel beta sheets, and then these anti-parallel beta sheets, after that, one of them actually completely twists on the other region to form this wedge-shaped structure. So you can see that it actually forms the floor, then the wall, and the wall. And these actually will interact with the next one and the next one the same way. And this topology is actually very, very useful to build these structures. So think about that. This is the block. This is the building block. And these, like again, in the 3 dimension, think about that. These structures are building the floor of this block. These structures are building the part of the wall of this block, and the other distant wall is built by this. Now, on top of this, there will be another protein which will help in contributing this part of the wall, this part of the wall, and the back, and this will go on to have the symmetrical structure to be built in. It is really very difficult to visualize in the 2 dimension, and it is not required either for the course. On the other hand, here you can see that it is also a very small structure, and these are the crystallographic structure of the 3 proteins that you are seeing. Don't worry about this one. This one is called the VP4, which is connected to the VP2, but this is inside the particle. It is not on the surface, and we're not considering this. These are the 3 colors, but remember that this color doesn't correspond to this color. So they are different, but these are the VP1, 2, and 3, and these actually built the poliovirus as such. So what will happen? Your VP1, 2, and 3, these will be in the quasi-equivalent position because these are not the same protein. If it is a VP1 one monomer, VP2 second monomer, VP3 third monomer, they could have identical interactions. But as now there are 3 proteins, these interactions need to be similar as per the rule of quasi-equivalence, but they are not. So what we mean by that, the interaction between the amino acids here, interactions in the amino acids here and here will not exactly the same. So they are similar to make this structure, but in the microenvironment of these conditions, they are different. So, but still the quasi-equivalent actually do exist, and that helps us to understand these structures very clearly. Now, this is also another very unique structure, SV40. Here, the T equal to 7. But according to T equal to 7, we should have 420 subunits. But there is not 420 subunit in the SV40 structure. SV40 is a polyomavirus that can cause cancer in some of the animals. And what you are seeing that instead of this virus is made up of 72 pentamers of the VP1. So you can just easily see that you multiply the 72 by 7, it should be, you should get basically, so technically, if you are T7, so your number should be 60 multiplied by 7, 420 should be the structural units. But in reality, there is just only 360 subunit made up of 72 pentamers. And how it is happening that suppose right here, you have this VP1 protein, which is interacting with 1, 2, 3, 4, 5 neighbors. But right here is you have a VP1 protein. All of them are VP1. Remember that. They are just the different structures of that. It is interacting with 1, 2, 3, 4, 5, 6 proteins. So again, carefully look at that. Here, it is interacting with 5 neighbors. In this position, it is interacting with 6 neighbors. So this is actually very difficult to explain because the same protein will have different interactions in different structural environment. And these are usually stabilized by the calcium ions and many other areas. And this actually opens up the surface of the SV40 a little bit. So compared to the poliovirus, which actually forms a very tight capsid, the structure due to this difference of this 360 subunits instead of 420 is more like a bristle-like structure. There is a little bit of opening in between these bristles. And here it actually shows that these interactions are not really by this beta barrel motif or the topology that you were seeing, but these interactions are more of the N and the C terminal regions of the individual proteins. The interaction is different. So everything is different for SV40. Beta barrel jelly roll structure doesn't work in here. So the next one is, again, even larger complex, which is the adenovirus. Lots of proteins. You do not have to memorize all the name of these proteins at all. Just that overview of that is good enough. So here is T equal to 25 capsid, and you have a 720 copies of viral protein 2, 60 copies of protein 3, and it is a very, very complex structure as well. So there are fibers at the 12-fold axis. You can see like there, and the presence of the proteins also devoted to specialized roles. What happens that due to this quasi-equivalence, as you can see, that as these proteins are being built, they are needed to be interacting with the neighbors in a different manner. But as these interactions are different, by the rule of quasi-equivalence, they has to be identical, but they are not. they had a tendency to fall apart or not stable. So there is a glue protein which is actually used, which is the protein 9 here, which is the cementing protein. And this actually stabilizes this penton and the hexon mismatches. So the same way that these are also, as these are larger structure, these are also made up of the pentamers and the hexamers. Remember that any structure which is more than T equal to 1 has to be built with the 6-fold symmetry as well. So they will be made up of pentamers and the hexamers. And here we are looking at a hexon trimer of the adenovirus protein. Just an overview of that is good enough. Do not have to go into very much detail as such. So here is a complex capsid with 2 icosahedral protein layers. And this is your reovirus. Very interesting structure. What do we mean by that? Remember that this is a naked virus. This is not an enveloped virus, but it does have 2 structures. You have a capsid outside, then you have a capsid inside. And here T equal to 13, 2 concentric icosahedral shell. But which is not shown, the rotavirus is also a family of this reoviridae. It's a double-stranded RNA virus with segmented genomes. And what happens that for the rotavirus, there's even another additional layer, like inside this, which is not shown here, inside this, there will be another icosahedral layer, 3 layers. And here you can see that for the regular reovirus, say, for example, a blue tongue virus, this is right here is the VP7 trimer. And here T equal to 13. And you can see that this is a 2-fold axis. This is the 5-fold axis, 1, 2, 3, 4, 5, you can see. And this is the 3-fold axis. You can see that these are 3 green, 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 3-fold axis, 2-fold, 2 blues right here. You can see. So right here, it shows the typical structure that how it is built. It's icosahedral symmetry, but again, very complicated or just roundish structure that how it has been seen. On the other hand, the inner structure, which is this inner structure, is made up of these VP3 monomers. Here, the T equal to 2. And you can see that right here, you have a 5-fold symmetry. You have 5 greens. Then you have a 2-fold symmetry. You can see that right here, either 2 reds or 2 greens across. And a 3-fold symmetry, you have this red here, red here, red here. So what is being shown that if you just take just a equatorial face or the structure, although the red and the green, they look that they are interacting in the same way, but they are actually not. So that is the very important structure that gives you the idea of the quasi-equivalence. So you can think that around the 3-fold structure, this protein actually covers, the VP3 actually covers as a plug, which actually helps to contain the genome inside there. And they will help in sometimes in the replication process that we will see somewhere else. So this is your complex capsid. So next, what we are going to look at, the tailed bacteriophages, and even more complex capsid. And you can see that right here, you have the head built with the icosahedral symmetry, where you can see that the proteins, the blue ones are called the GP23. The purple ones are interspersed in there, which is called the GP24. And these yellow ones are the spikes coming out of that. And the white proteins are the cement proteins in there. So you can easily see that these purple proteins in there and impregnated in there, the GP24, actually does not follow the rule of quasi-equivalence. So the thing is that it just gives you an idea to kind of understand the structure, but there are lots of anomalies in nature which doesn't follow the rule of mathematics. And that's how these structures are being built, but still they are very stable. So here you have the head, and then you have this connector and the collar region. Then you have this contractile tail protein right here, which is built with the helical symmetry. So you have the head with the icosahedral symmetry, and this with the contractile tail with the helical symmetry. And some of them are contractile. So what happens that these long fibers, they actually attach onto the bacteria, and this base plate actually is inserted onto the cell wall to bore a hole. And this whole of this contractile, the tail or the neck, this protein sheath will compress, and this will cause a huge amount of pressure built inside this head, and that will inject the DNA inside the virus. So you can easily see not only the protection, but also the delivery of the genome that we did not talk about the other places. But here you can easily see the delivery of the genome is also shown by the capsid protein, which in the very first two slides we observed. The next, what we are looking at, a very complex virus, which is a herpes simplex virus. This does have a very unique structure. And what happens that there's a portal or opening of the viral DNA is built on one of the 12 5-fold vertices. And here, this is a T equal to 16, 200 nanometer in size, large, very large herpes virus, double-stranded DNA virus. And what you can see that this is just a slice of the tomography, cryo-EM tomography that you can see. And right here is, although you cannot, the image analysis needs to be done by the computer, you cannot see, but right here is the portal. On the other hand, this is very interesting right here. And this is an immunoelectron microscopy where the portal protein, which is known as the EU7 or EUL7, that is being conjugated with a gold particle. And if you have taken cell biology, you already know that how you do this immunoelectron microscopy with the gold particles. And these two dots actually tells you that where this portal is. So think about that. That does not give you any symmetry. This is built on the icosahedral symmetry. The nucleocapsid right here is the inner part. And then on top of that, you have the membrane. But just there is only one part where you have a portal like this, which is used to package the DNA inside the particle, as well as it helps the DNA to come out once it infects the cell, beautifully shown here. So you can see that definitely this is a very asymmetric looking capsid, or the envelope on top of the capsid. So the capsid, as we said, can be covered by the host membranes. The envelope is a lipid bilayer derived from the host cell machinery, and viral genome does not encode the lipid synthetic machinery. The envelope is acquired by budding of nucleocapsid through a cellular membrane, can be any cell membrane, but is virus specific. And the nucleocapsid inside the envelope may have helical or icosahedral symmetry. So if we just go back right here in this picture, you can see that this nucleocapsid does have an icosahedral symmetry. This is an example right here. This is an example of the HIV. Not HIV, this is a cartoon of a retrovirus, and HIV maybe because there are two identical plus RNA molecules, so it's very specific. So this is a retrovirus, and you can see this inner core is helical compared to that. So the next thing that on the surface that you are seeing, an example of influenza right here, the cartoon, there are glycoproteins, and we know these glycoproteins very well. For the influenza, these are the H and the N's, which are the hemagglutinin and neuraminidase that you are seeing, which is sticking out of the membrane. And these are the ectodomains, which are the outside of the membrane. And these ectodomains, which are sticking out of the membrane, are used for attachment, antigenic site, or the fusion. On the other hand, the internal domains, which is right here, they are help in the assembly structure. And sometimes they can form oligomeric, means there are multiple, like a trimers, tetramers, dimers, and they can sometimes form spikes as well. So here is a typical structure that you are seeing. That's right here is the internal domain. Then you have a membrane spanning domain, which is mainly alpha helices. And then you have the ectodomain, and you can see that all these different structures are stabilized with the disulfide bonds. So the greens are the disulfide bonds, whereas these purple Y-shaped structures are the carbohydrates. So these are mostly the glycoproteins. So let's take a look at some examples right here. This is right here is a glycoprotein from the influenza virus. This is a trimer, and only just one is shown in the red and the blue. So right here, this portion is the globular head. This actually binds to the receptor, and this is a hinge region stem, and that is inserted into the membrane. This is the lipid membrane. This is the envelope of the virus. On the other hand, this is just how it looks like. So this blue ones right here, these are your influenza virus spike proteins. But on the other hand, another member, flaviviridi virus, This is an example of a protein which is on the surface, which is not a spike protein, but it is a flat structure. So you can see that this structure is made up of two dimers, two different colors, dimers of a protein, and they actually lay flat on the surface. So very different from the spikes. So the envelope can come in all different shapes and sizes, not necessarily the glycoproteins on the envelope has to be spiked all the time as well. So next we are looking at some of these complex virus particles. You can see that the pox virus, and there is always a controversy about the pox virus, that whether they're covered with a single membrane that most of the envelopes are, or where there is a dual membrane that we see sometimes. But they actually do have a core which is surrounded by the membrane. It is so large, there is no cryo-electron microscopy beyond this resolution is actually available. We still cannot do that. On the other hand, here you have the Mimi virus, which actually shows icosahedral symmetry. This actually doesn't show any symmetry. On the other hand, this shows, except that there is a portal that we saw in the herpes virus at one vertices, but other vertices have these fiber-like structure. So this pithovirus or the Pandora virus also are built with this helical symmetry, and there is a portal of entry or right here for the genome as well. So you can see that they can come in all different shapes and sizes, and we are still understanding the symmetry of these giant viruses and the bigger viruses. On the other hand, what are the different virion components and why I enlisted that under this structural biology? They are polymerases, integrases, associated protein, proteases, polyopolymerase, capping enzymes. Some of them are activator proteins, some of them are cellular components. And why they're important in the sense, and this is an example that has been shown in an HIV molecule, that what are the different proteins in here. Envelope protein, capsid, matrix proteins, reverse transcriptase, protease, integrates, and the nucleocapsid protein. Now, some of these proteins right here, the enzyme activators and the cellular components, can be a structural component as well. And if they're the structural component, it is logical to talk about them here. But there are some times, say for an example, if we just look at this protease structure or the reverse transcriptase, they do not have much of the integrase, they do not have much of a structural role on the viruses. So then they are called the non-structural proteins. So some of these could be structural as well as non-structural proteins. So that's pretty much this chapter. I know that I just went a little bit fast in this context, but any questions so far? If not, I will quickly show you one quick thing. Let me do. How much time do we have? We have 2 minutes. Okay, so share screen. Okay, you probably can see my desktop. All right, so here is a program that we use to build three-dimensional structures. So this is called Blender. This is free. It's an open source software. You can download it. It's a very steep learning curve to understand that, but you can look at the structures for fun purposes here in here. So here I just drew a simplest polyhedra. And now this will help you to understand the 5-3 and the two-fold axis. If you just look at this structure right here, if I'm looking, take out the camera right here. If I'm just looking at from this direction right here, you can see that right here, this where I'm pointing out right here, this is your 5-fold because you have 1, 2, 3, 4, 5 triangles around that axis. I'm just moving it in another direction. Say for an example, right here, if I'm just centering this polyhedra right here, then you have one triangle on this side, another triangle on this side. So this is your two-fold axis. And then, of course, if I just point it right here, I'm in the middle of the triangle, and this is your three-fold axis. You can see that one triangle here, one triangle here, one triangle here. So this is your three-fold axis. So this gives you an idea of that how in the 3 dimension these three-fold axes actually work. you can actually go beyond that. And if you just make these subsets, we do not have time, it's 9:15. But if I have built the triangulation number, this icosahedral structure, you will see that there will be another structure will actually develop that instead of this pentameric assembly, you will also have an hexameric assembly in there as well. So, if you can just look at that, this is your 1, 2, 3, 4, 5. But you can see that right here, if you just look at this point right here, you have 1, 2, 3, 4, 5, 6. So here you have a 5-fold symmetry in this position as well. So this is just to give an idea that how these 5, 3, and 2-fold symmetry actually does work. So that's pretty much about it, I guess. So you guys good? Any questions so far? I know that it may you may feel that it is a little bit intense, but I have put extensive note in there. There's just a couple of spelling mistakes, so download those, and I will just correct that and upload. Just rewrite that file just one more time, as such. Any questions, folks?
I had one question about the spike proteins.
Yeah.
So you said that viruses don't encode the envelope, and they kind of like steal that from whatever cell they're infecting. Do they encode the spike proteins, or do they?
Yeah. So the thing is, a very, very good question, actually, Matthew. I appreciate that. So all the proteins that you are seeing in the membrane, those are all made by the virus. And the way it works, that they stay very closely associated with the inner capsid. Then, while the virus actually exists, they actually get the membrane from either the ER or the nuclear membrane or the plasma membranes. And at that time, for a short period of time, the top of that membrane is completely flat or plain. This is an immature virus particle, and it does not still is infectious. Once this virus buds off from the particular area, these proteins, which are the spike proteins, no matter what they are actually travel through the plasma membrane by complete chemical interaction. We will see that actually, particularly through the areas which we call as a lipid rafts in the membrane. There are some proteins have a specific affinity for these lipid rafts, and they actually stick out of the membrane. And once these are put into the membrane and stick out of the membrane, that becomes a mature virus particle, and it becomes infectious. So it is made with the proteins packaged close inside the virus particle, initially remains underneath the membrane. When the virus matures, these are transported through the membrane and sticks out. Does that answer your question?
Yep. Thank you very much. Yep.
Yeah, we will talk about that in very detail. Any other questions that you may have?